Good morning. Today, inshallah, we're going to talk about microbes, part one. First of all, what are microbes? We can also call microbes microorganisms. It comes from the word tiny or small. So, microorganisms mean a very small organism that you cannot see with your eyes and you need a microscope to see it with. So, microorganisms or microbes are small living organisms that you're not able to see with your eyes and you need a microscope to see it with. Here is the light microscope. In the last session, we revised the labels of the light microscope that we use in class. We start looking. Take a quick review on that. First of all, you have to adjust the objective lens, the low power objective lens on your slide. Then you're going to look from the eyepiece lens in order to investigate the slide. At first, you're going to turn the coarse focusing wheel until you see an image. After you see an image, you're going to start to turn the fine focusing wheel until the image is clear. At this point, you're going to see your slide. After that, you can switch to the medium power objective lens, and you're going to continue with the same sequence. First, turn the coarse adjustment wheel until you see an image, and then turn the fine adjustment wheel until you see a clear image. A clear image for you shouldn't be clear for your friend or for someone else, so you may need if you're investigating a slide after someone, you may need to change the fine adjustment wheel so that the image would be clear for you too. Okay, what is this? This is the microscopic stage where you put on the slide, and this is a clips to fix the slide on the microscopic stage. You can use a mirror or a light source in order to let light pass through the slide to be able to see it or to investigate it. Okay, we have four types of microbes: algae and protozoa, fungi, bacteria, and viruses. First of all, the algae and the protozoa. The algae, this is how it looks under the microscope. Um, the algae, we call them a very tiny plant-like structures. Why are they like the plants? Because they contain chloroplasts and they are able to make their own food by photosynthesis process. But the protozoa, they are like the animals. This is how they look under the microscope. One example of protozoa is the amoeba. They live in the water and they feed on other microorganisms from the water, or they may feed by absorbing nutrients from the water. Like, for example, they may feed on um, dissolved solid waste in the water. The main types of microbes and the most important ones are the bacteria, the virus, and the fungi. The fungi are the largest, the viruses are the smallest, and the bacteria in between them. So bacteria, viruses, and fungi. First of all, the bacteria, it's a unicellular organism or a single-celled organism. This means that the organism is made of only one cell. Some of them are useful and some of them are harmful or pathogenic that causes disease. We have a lot of shapes of bacteria like the of bacteria like circular, rod, or spiral shape. As you see in this slide, those are different shapes of bacteria. Sometimes the bacteria, it can live on its own. Sometimes it lives in a pair and sometimes they live in colonies. The singular of bacteria is bacterium. So one bacterium, a lot of bacteria. Concerning the cell structure of bacteria, this is the whole organism because as we said, the bacteria is single cell. Uh, the bacteria is only made of only one cell. Uh, it has a cell wall and a cell membrane, and the genetic material is loose in the cytoplasm. It's not covered with a nucleus, so we don't have a nucleus in the bacteria. The genetic material is present in the cytoplasm. Sometimes we have an extra membrane in the bacteria, which is this blue part. It's called the capsule. It gives extra protection. Those are the labels that we need to know. In the exam, if you have only two membranes, those will be the cell wall and the cell membrane because not all bacteria have a capsule. As you see here, what is this? This is a cell wall and this is the cell membrane, and we have here the cytoplasm and the genetic material or the chromosome. You don't have a nucleus. The genetic material are present in the cytoplasm. The same here, the same labels. So as you saw here, there isn't a capsule in the bacteria shown in the previous two slides. Here is the bacteria dividing. It divides by a process called binary fission, where one cell divides to make two, and then the two makes four, and so on. Of course, this is much faster than the normal rate. It's 600 times faster. Actually, the bacteria reproduces fast. It can reproduce every 20 minutes. So if you have one bacterium, you're going to have two after 20 minutes. If you have two, you're going to have four, because each one is able to divide into two every 20 minutes. Okay. Uh, then regarding the virus, a virus, it's not living because it's not able to reproduce except inside a host cell. A host cell, it is the cell of the organism that the virus infects. It can be a human, an animal, a plant, or even a bacteria. So we don't consider that the virus have cells. It's just the genetic material inside a protein code, because it's not living, or it's a link between living and non-living organisms. Why it's a link? Because it is not able to reproduce on its own. It needs a living organism, a host, to reproduce inside its cells. So what are the labels of the virus? We have a genetic material inside protein code, and here we have the head and the tail. The most important thing is to write the genetic material inside the protein code. Concerning the fungi or the fungi or the fungi, the three are correct. The singular is fungus. Some of them are microscopic, are microbes. Others are not microscopic, and you're able to see without a microscope. We call them macroscopic fungi, like the mushroom. The fungi shows all the seven characteristics of the living organisms, and they reproduce by producing spores. So here are the spores. They get out of the fungi and they spread in the air, and when they land on suitable environment, they grow to form a new fungus. It's something similar to the seeds in plants. This is the yeast cell, a type of fungi. The yeast is also unicellular, or a single-celled organism, but not all the fungi are unicellular, only some of them, like the yeast. It has a cell wall, a cell membrane, a vacuole, cytoplasm, and nucleus. Its structure is very similar to the plant cell, except that they don't have chloroplasts because they don't need to make their own food. This is how the yeast reproduces. It reproduces by a process called budding, where the yeast makes a bud, then the buds grow and split. Sometimes uh, the bud remain attached, and the yeast produces more buds, and the buds grow, and those new yeast will make other buds, and all the fungus remain attached to each other. They don't detach like this one. So it depends on the type of yeast. Okay, here you have, this is the bud. Of course, this is the cell membrane, the nucleus, the cytoplasm. We don't need to know this, which is the food storage granule. We need the vacuole and the cell wall. So we need to know all the labels, but we don't need to know the food storage granule. We can see those slides under the microscope. Here is the bacteria. You can magnify it to see the circular or the rod shaped bacteria. Here are the fungi. This is the yeast. As you see here, they remain um, attached together, and some of them also may split. Here is a type of virus. Okay, a macroscopic fungi like the mushrooms. The mushrooms also reproduce by producing spores. The spores come out from the gills, which are under the cap of the mushroom. The spores just fall down near the parent mushroom and grows to make other smaller mushrooms beside the parent one. They have a stalk and a cap. Inside the stalk and below it, we have hyphae. It doesn't go down or deep in the ground. It's just about 5 centimeters deep, not more than that, but it's branched a lot. Most of the time, the mushroom feeds on the dead wood or the dead leaves or twigs. So they get their nutrients from the dead wood or leaves or twigs. And when they consume the nutrients, they can branch more and more to get the nutrients from more twigs and more pieces of wood. Here is a mushroom. As you see, it grows where you find the leaves and the dead wood. If you want to grow the bacteria on a petri dish, as you see here, we can see a colony of bacteria, a lot of bacteria. As we said, singular bacterium is not seen under the microscope, but here I can see a colony, thousands or more of bacteria. That's why they become visible, because it's not only one. How would you cultivate this bacteria on a petri dish? First of all, you're going to get the petri dish. It has to be sterilized. Then you're going to put the agar. Also, the agar has to be very hot and sterilized. And afterwards, you're going to use a cotton swab and get the bacteria from where you want, for example, from a mobile phone or a remote or anything that you need to uh, see the bacteria that grows on. And then with a cotton swab, just go over the agar a lot in all the directions, and then close the close the petri dish carefully with another lid and tape it, and then leave it in a warm temperature for 24 hours and observe the growth of the bacteria. Why did the bacteria grow? Because you provided for it two things: suitable temperature, which is warm temperature, and also you provided for it the nutrient, which is the agar that the bacteria feed on. That's why the bacteria grow, and you're going to have a lot of bacteria, and it's going to be visible for you. Okay. Uh, we have to make a lot of precautions while doing this experiment. First of all, you have to wear gloves, masks, and goggles, so that no bacteria can harm or enter your eyes, or you won't inhale it through your respiratory system, so you need to wear the masks. Also, the lab bench should be clean before and after the experiment. Before the experiment, because I want to see the bacteria only from the remote or from the mobile. I don't want bacteria from other sources. So I don't want, for example, to get bacteria from the bench. So I need to clean it first with a disinfectant, like Dettol, for example. Also, you need to wash your hands before and after doing the experiment, and you need to tape the petri dishes well. Okay, now we're going to talk about decay. What is decay? It's the breakdown of the decomposition of organic matter by microorganisms into simpler forms. The organic matter is any substance that is made of living organisms. It can be a dead organism, it can be a part of an organism like a leaf or a twig, or it can be the waste of the solid waste of organisms. Okay, a decomposer, it's a microorganism that causes decay, that breaks down the organic matter, which can be a dead body, part of the dead uh, body, or the waste of living organisms into simpler forms. Okay, decay can be useful or it can be harmful. Useful because it prevents the accumulation of dead bodies, and also it returns the useful nutrients back to the soil. Harmful in case of rotting of fruits or in case of tooth decay. Those are examples of harmful decay. Okay, how do we use bacteria in food production? First of all, we can use bacteria to make yogurt and a lot of cheese. How do the bacteria make yogurt? Um, a lactic acid bacteria or a lactobacillus bacteria feeds on the lactose, which is the milk sugar. the sugar in the milk is called lactose. It feeds on the lactose and uses it in respiration. Respiration is the breaking down of food. So the bacteria breaks down the food, which is the lactose, to get energy. With energy, lactic acid is also produced. This lactic acid causes or leads to two things. First of all, coagulation of milk protein. So this changes the milk from liquid to solid or semi-solid, and it also gives the yogurt its sour taste. Bacteria is not only used to make yogurt, it's also used to make antibiotics, cheese, certain kinds of wine, compost, blue cheese. It can be used to produce fuel. Okay, we can also use other microorganisms in food production like yeast, which is a type of fungus. As we said before, it's a unicellular or single-celled fungus. Also, the yeast is used in bread making, and it is used in making of beer or brew. How? By two different methods. Here, the yeast feeds on the sugar glucose that you put in the dough. Uh, if there is oxygen, so the yeast will feed on the sugar glucose and use it in respiration. It will break it down in the presence of oxygen to produce energy. With energy, water and carbon dioxide are given off. The carbon dioxide is the gas or the bubbles that will make the bread dough rise. This is called aerobic respiration because it happens in the presence of air or oxygen. If the respiration was anaerobic, if there is no oxygen used, this will lead to the formation of carbon dioxide with ethanol instead of water. And this ethanol is alcohol that we use to make beer and wine. This process is also called fermentation or is also known as fermentation. So here the yeast will use the sugar glucose will feed on it and use it in respiration without the presence of oxygen to produce carbon dioxide and ethanol that is used to make the beer. Okay, so that was it for this lesson. Hope that you understand it. Thank you and see you next time, inshallah. Thank you very much.